Alrighty, so we are going to take a look today at a process called DNA replication. And when you first think of the word replication, you need to think of the word replicate, and that just means to make more of. All right, and that's basically the whole point of this DNA replication. All right, so we start with one strand of DNA, and we're trying to make it into two. All right, so we are taking a look at your notes. This is starting on page 76. We're going to real quickly go through those, and then I'm going to illustrate out this process as well. All right, so DNA replication is the process of making more copies of DNA for cell division. All right, so we got a little recycling sign here. It's just showing us once again that we are making this DNA copies for cell division. So if we think back, we learned about the process of interphase, which is the normal life cycle of the cell. All right, so during that interphase, let me try to get this paper. There we go. All right, so during interphase, let me find a pin that works. All right, this normal life cycle of the cell, we have a couple different components. All right, so we've got G1 and G2, which is standing for the growth of the cell. So the cell is growing, all right? And then they have something referred to as S phase, all right? S phase stood for synthesis, and we know that synthesis means to make, all right? So during this phase, we are actually making more DNA. It's when our DNA is being copied, okay? And that is the process of DNA replication, all right? So because DNA is so important, when a cell divides, it has to create an exact copy of the DNA in order to function correctly. Therefore, that DNA is replicated, once again, during the S phase of the cell cycle, which is a part of interphase. It's all before mitosis and meiosis can begin. All right, so the process of DNA replication is actually pretty simple, all right? So we've got a couple different steps. We've got steps 1, 2, 3, and 4 laid out for you. All right, so I'm going to illustrate those as we kind of follow along in the notes themselves. All right, so the very first step of DNA replication. All right, so our very first step of DNA replication, I'm gonna zoom out just a little bit so that we can see that. All right, so we've got our DNA strand. Remember that it's forming kind of a double helix structure, two-sided strand. All right, so step number 1 is that an enzyme comes in, and an enzyme breaks the hydrogen bonds. Remember that we learned that hydrogen bonds are weak bonds, and that's what's holding those those nitrogen bases together. Sorry, not nucleic acids, those nitrogen bases together. So if we think back to our DNA structure, remember we said that it has a sugar phosphate backbone. I'm going to symbolize that by showing S and Ps as we go down the backbone side. All right, and then in the center, we have those nitrogen bases. And if you think back, we remember that A, adenine, and T, thymine, will always pair together, and C, cytosine, and G, guanine, will always pair together. All right, so if we copy those, that's what our DNA strand is going to look like. All right, so that enzyme is going to come through, and that enzyme is actually going to break all of these hydrogen bonds, creating basically almost a split DNA. All right, so we're going from this to 2 separate strands of DNA. Now notice that I'm keeping my letters the same because the letters have not changed. Those nitrogen bases have not changed and the order has not. All right, so just those bonds are now broken. All right, the second step is when these new unpaired nucleotides start to come in and attach to those strands. All right, so we've got these strands here. I'm gonna keep them the same colors. All right, so we've got our left-sided strand, which was A, C, T, and C. So A, C, T, and C. All right, and our right-sided strand was T, G, A, and G. All right, so now these new unpaired nucleotides are coming in and they're starting to attach to these open ends. All right, so if we have a new unpaired nucleotide, this nucleotide that's going to come in, if this is adenine, this one should be thymine. Okay, if we have this one, cytosine, it should pair with guanine. T should pair up with A, and C should pair up with G. All right, so let's do this side now. We've got an unpaired nucleotide. T always pairs up with A. G always pairs up with C. A pairs up with T. And G pairs up with C. All right, so then it's completely proofread the same way that you would proofread a paper before you turned it into your English teacher. And I want you to notice what is the result of this particular DNA replication, okay? So these are your two products. All right, and this is the way that we're going to describe them, all right? So we are going to say that our new 2 copies of our DNA, our 2 copies are identical copies. Notice that they both go ATCG, TACG, ATCG, TACG, okay? All right, each strand is half old DNA, half original DNA, and half new DNA. Okay, so half old, half new. So notice that the blues were representing the original DNA strands, and the pink was representing the new side of the DNA strand. Once again, half was original, half is new. All right? So we refer to this as, and this is a great term to know, this is called semi-conservative, okay? And semi-conservative just means semi is somewhat or half, conservative means staying the same. Okay, so half was the same, or half was the original DNA, all right? So each copy of that DNA is then all packaged up as a chromatid on a doubled chromosome, and then meiosis can occur or mitosis can occur. So now we've got these 2 wonderful copies of DNA, and we can go through the process of cell division.